yeah, uh, today I'm speaking from my apartment. Japan is already night, and so I'm from East, and today so I'm going to talk about spontaneous activity in spiking neural network. And so before I forget, all what I want to talk is collaboration through with these three uh, postdoc and one student. So yeah, they already left my lab, but yeah. Okay, so basically, so I want to talk about the two models that are mutually kind of related, but slightly different topics. And so both are related to spontaneous activity or spontaneously replay. So before going into detail, I want to discuss why I'm interested in spontaneous activity. So why spontaneous activity is interesting, at least to me, and important. And first of all, so it is an inherent dynamical feature of the brain, but not of artificial machines. So as you may know, a lot of processes are going on during spontaneous activity. So understanding the functional role and mechanism of this will be crucial for understanding the brain function. And so experiments show that spontaneous activity, so basically replay, is during sleep and awake is crucial for memory consolidation, decision making, and planning of future action and inference. So spontaneous activity is related to many cognitive functions. And specifically, so spontaneous activity represents an optimal model of the probabilistic external world. And this related to the first topics. Yeah. Okay. So in my first topics, what I specifically want to talk about is about so motivated from this very famous probability experiments. And so what these people did is they show some visual images to animal ferrets and observed what kind of activities evoked in visual cortex. And in some cases, they show just blank images and observe the kind of spontaneous activity without any visual stimulus. And what they found is after maybe 3 months or something, so actually activity evoked, spontaneously evoked in visual cortex is a superposition of activity evoked by visual stimuli. And they actually found these states represent optimal uh, probabilistic model of sensory experiences. And this triggers actually many modeling studies that not many models are literally modeling spontaneous activity. And this is why I was interested in these topics. And why so spontaneous activity has to represent the optimal model? So according to Bayesian arguments, so some process is going on in the external world, and observation is given from this process. Then brain kind of assign some states, internal states, and basically brain try to infer what is a given observation by using Bayesian formula. Okay, so the spontaneous activity here will provide so-called prior distribution, yeah, of environment, yeah, between this blue. And so the fact I want to model this, yeah, using spiking neural network. But I think at least this process contains at least 3 steps. Yeah. So first, we have to segment statistically salient stimuli from the stream of stimuli. Yeah. So generative process in the external world produce a sequence of stimuli continuously, and brain has to know or segment which input is important, at least statistically, meaning which input appears more often than others. Then the brain has to generate maybe kind of stimulus-specific cell assemblies to encode these salient stimuli. Then third process will be when stimulus is not given, brain should replay these memory assemblies at the probability these stimuli occur. And there will be a lot of different ways to approach this problem. But what I want to suggest is at least the first computation can maybe done by single neurons. You do not need actually, you may not need a complex network. Yeah. So how this can be done? So for this, yeah, some years ago we proposed so-called predictive learning or self-supervised predictive learning. So the basic assumption is dendrites learn to predict somatic response in a self-supervised manner. Yeah, I explain what I mean by this self-supervision. Yeah. さあ、10名の、so this is a pyramidal neuron. And pyramidal neuron fires back spikes, then spikes also propagate backwards towards the dendrite. And yeah, in some cases, this back BAP is important for learning. So what we did was we basically model or mimic this effect of back propagating action potentials in a mathematical way. So this is a learning rule. So yeah, if you are familiar with machine learning, this means so single neuron is receiving synaptic input, and so you have some dendritic activity, and dendritic activity try to model somatic response. This one, and so from in machine learning language, it means spike density of soma is modeled by dendritic activity. So KL divergence between two density function has to be minimized. And so learning at dendrite occur according to this law. And of course, dendritic activity is given by synaptic input. And so this might look a bit mathematically complicated, but actually what we obtain, so by calculating this, we obtain some Hebbian-like plasticity. So this is somatic spike density, and this is dendritic activity. And so this is a postsynaptic activity. And E is EPSP generated by sensory input. So this subtraction looks slightly different from Hebbian, but basically this is a multiplication of postsynaptic activity and presynaptic input. And we basically average if there might be many different input patterns. So input comes, some salient stimuli comes in sequence. So input contains multiple interesting sequence might be. So if this quantity has to be averaged over input, and also this is quantity is averaged over sufficiently long time. That's T. Okay. And in principle, if this works, so what we found is something. Oh, okay, yeah. Maybe it's better to first show you example. So here, this is single neuron, simulation of single neuron. So single neuron is receiving 2000 synaptic input. and each input is just a irregular spike train. And there, and so these black ones are just random input, and these they never are repeated. However, this green one, blue one, and red one is irregular patterns repeated, intermittently. And if this pattern input is given to a single neuron, then initially the neuron respond to, just so much generate response at arbitrary moment. However, when learning proceeds, then in this particular example, this neuron started to detect red patterns. Yeah. So all other patterns are ignored, but this is neuron selectively responded to red pattern. And why this occurs is, basically dendrite try to predict somatic response. But that is possible only when there is some regular patterns in the input. So if dendrite started to receive respond only one of the regular patterns, then prediction is possible. And for doing this, there is one mathematical trick. Actually, is if so you have to kind of normalize, standardize the dendritic input to the soma. So sufficiently long time, you have to always standardize input, and this can be done by shifting threshold and slope of response function of the soma. And why this is necessary is if so, this basically keeps dynamic range of dendrite within certain range. So without this, then what happens is neurons basically learn the trivial solution. So all weight becomes zero. And this is actually one of the solution of our learning law, because if W equals zero, then there is no input, so there is no response. So the prediction is perfect. So this is obviously not interesting. So our model answers, so dendrite, soma supervised dendrite, but so without any external signal. But to do this, so you need to normalize a dynamic range always in a proper range. Okay. So this contrasts with a similar model for supervised learning where soma was receiving external input. In this case, you do not worry about trivial solution. Okay. Okay. So I want to use this learning rule for probability learning. So here I have actually three compartmental neuron. So this is a network of three compartment neuron, and we assume all in synapses, meaning so there are afferent input, recurrent excitation, and recurrent inhibition. But all these synapses try to predict somatic response. Okay, so we use the same learning rule. And this actually allows this network to learn probability of afferent input. Okay, so so first of all, each neuron. So there is input cells, and initially each input neuron projects to all these neurons in the reservoir. Okay, so there is no each neuron in the reservoir doesn't have any stimulus specificity. So the first segmentation should occur. And so this is the example of simulation. So now the network receive 5 repeated input patterns with different probability. Okay, then so initially neuron doesn't respond. However, neuron start to pick up one of these patterns And yeah, so response is gradually generated. And after sufficiently long time, oh, sorry, something. All right. Then each neuron in the these of us selectively respond to one of these stimulus. So then at this moment, segmentation and also because recurrent connections are formed within these neurons, so segmentation and formation of cell assembly is done. Then the next question is whether this evoked activity is replayed with the occurrence probability of each stimuli. And yeah, so this is a spontaneous activity replay. So you can see here, so each pattern actually is replayed. And if you plot the occurrence probability, so this is a probability, relative probability of assembly, this one. And actually firing rate, so population firing rate follows this given probability. And if the probability distribution changes, then the probability that each stimuli is replayed are also changing. And actually, in this model, so what I mean by population firing rate is you just count all the spikes within the cell assembly responding to the same stimuli. Okay, yeah. Assembly size and the firing rate also changes, but not accurate. These quantities do not accurately represent probability. But why this is possible was so basically, so each neuron when they learn stimulus try to so try to learn the probability of firing of somatic firing. So because all recurrent connection also run that probability distribution. So even without the stimulus, um, they can spontaneously replicate what they experience. This is the underlying mechanism. Okay? And this is a bit too detailed, but later we introduced, so in the previous model, excitatory neuron and inhibitory neurons are not separated. So we introduced separate excitatory neuron population and inhibitory neuron population, and basically applied the same learning rule. But actually, we struggled, and what we eventually found was if excitatory neuron and inhibitory neuron are separated, then we need at least two types of inhibitory pathway. So one is operating separately with excitation, but another path two, operate together with excitatory neuron. And what happened is, so we separate, so we calculate this weight strength of within assembly and between assembly inhibitory neuron, inhibitory synapses. So path one, so between assembly inhibitory connections are almost zero or very weak. So it means path one probably is operating only within assembly. So actually, this inhibitory connection is necessary for decorrelate spikes within assembly. So so as a result of decorrelation, assembly, so firing rate of assembly has some function meaning. On the other hand, pass in contrast, so pass both within assembly and between assembly synaptic weights of inhibitory connections so equal magnitude. So it means, so because of between assembly connection, so this introduces kind of lateral inhibition between assemblies. So this complicates it. So the necessity of this complicated inhibitory circuit made separation of excitatory neuron and inhibitory neuron bit difficult. Okay. And I skip that. So we tested whether our model can replicate decision making of monkey. So here, right? So maybe many of you know this experiment that the monkey has to decide the direction of coherent movement of random dots. And if in training, left hand movement and right hand movement appear equal strength or equal probability, then there is no bias. However, if, for instance, left-hand movement appear more often than right-hand movement, then decision making is biased. Yeah. So more we tend to consider that. Okay. Anyway, right movement occurs more often if training has bias, and our model actually replicated this just very nicely. Okay, so I want to move to second topics, which is again spontaneous replay. But this is about memory replay. Yeah. So, so replay is important for memory consolidation. So during sleep, so this is a kind of schematics of widely believed hypothesis in memory encoding. Or episodic memory encoding. So during wakefulness, stimulus come to, then this is the hippocampus and this is neocortex. And the stimulus information is given to the hippocampus and maybe some assemblies are formed. Then during sleep, these assemblies are replayed and this information is sent back to neocortex and maybe some corresponding assemblies are formed in the neocortex. And actually, this represents long-term memory. And during wakefulness, when particular memory should be retrieved, then these neocortical assemblies are activated. And so this is what we currently believe. And as you may know, so Hopfield some years ago received the Nobel Prize. So what he proposed was, so he explained one characteristic of memory retrieval by using very simple neuron network model called associative memory model. So basically, you embed multiple memory patterns or binary memory patterns by using Hebbian connections into this network. And basically, each neuron is doing kind of thresholding and also calculated sum of input from other neurons. And if this connection is symmetric, then you can show that this network has attractor dynamics. And by using local Hebbian connection, you can make each memory pattern one of the attractor states. So depending on the initial state of the network, maybe initial state is set by stimulus, then even without stimulus, the network start to retrieve this pattern by using downfield motion along this energy landscape. So this Hopfield network actually attractor memory idea proposed by Hopfield actually influenced a lot about in neuroscientists. Yeah, but as you may know, or you may not know, actually I was a physicist. Yeah, Hopfield network has memory capacity. So, so later physicists shows that if the ratio between number of patterns and the size of network is within below critical stage capacity, then there is a successful retrieval case. However, if you embed more patterns than stage capacity, then no retrieval occurs. Yeah. So basically, these memory attractors disappears, and this occurs actually very suddenly. Yeah. So there is a phase transition phenomena. Yeah. And this is one kind. Yeah, people say this is kind of catastrophe forgetting in associative memory. So we wanted to consider this by using spiking network. And actually, what we think is there are no need of attractor states. Yeah. So what the hippocampus is doing is, so hippocampus has to replay memory patterns, but these replay patterns experimentally is shown just transiently retrieve. So we hippocampus need not be at a stable attractor network. So we kind of make a Hopfield network using spiking neuron. And E2E connections are generated by local Hebbian learning, but we also we used to we changed we modified connection matrix so that it obeys log normal distribution, because it is known that hippocampus weight distribution obey log normal. Yeah. And we also introduced inhibitory plasticity in I2E connection. Yeah. Here only excited neurons are drawn, but this network has both excited neurons and inhibited neurons. And I2E connection obeys plasticity. But we consider two types of synapticity. One is local balance rule. So this learning rules try to set the firing rate of target postsynaptic excited neuron at 3 Hz. And another is global balance rule. Actually, this was proposed by one of the organizers. And this try to set the firing rate of entire network at some level. Okay. And we studied how replay occurs, yeah, similarly or differently. So actually, thanks to inhibitory plasticity, so if after training, so the network started to show spontaneous replay of pattern embedded in excited network. So I just show 5 example, 5 most frequently replayed assemblies. Yeah. So assemblies are replayed kind of transiently and intermittently. Okay. And this occurs for both learning role. However, there are interesting difference. So we tested, first we tested whether pattern completion occurs. So we applied external stimulus or some cue to retrieve embedded pattern. And for if neuron or inhibitory plasticity is local one, then actually retrieval occurs, better pattern completion occurs. Yeah. Pattern completion occurs for both cases, but quality of retrieval is increased if we use local inhibitory plasticity. Okay. And this is a number of patterns embedded. So this is a range of patterns we embedded. I forgot to say. So the whole network has 8,000 excited neurons, and each cell assembly has about 80 neurons. Okay. And we embedded 85,000 assemblies in the network. And another interesting difference is, so this is the number of patterns replayed during spontaneous activity. So for both cases, some patterns are retrieved, but if I assume local EI balance or local balance rule, then network replay more patterns compared to global balance law. And this is, yeah, so we just increased the number of embedded patterns and counted how many patterns are actually replayed within 2000 seconds. And again, local balance rule. So both number of patterns decays. However, decays much slower if I use local learning rule. Okay. So it means local learning rule kind of replay diverse memory patterns, while global learning rule kind of selecting patterns, which patterns are replayed are selected by some mechanism, which we have to study very detailed. Okay, so this difference appear. And I'm already almost done. So we were interested in why these two differences appear. And so for doing that, we kind of linearized our model. Yeah, and so we kind of generated, constructed rate coding neuron model, and the synaptic weights are kind of linearized. Yeah, now, neural response was linearized, and we defined effective coupling constant. And we studied the eigenvalue distribution of this coupling constant. And so what happened is when number of patterns embedded are small, then two new network models shows similar distribution. However, if pattern is increased, so here, so for blue one means local learning rule, then still eigenvalues are within kind of circle. But for global learning rule, some eigenvalues started to deviate, go outside of this circle, meaning these actually states becomes unstable. Yeah. So more assemblies are spontaneously played when infinity plasticity is tailored assemblies specifically, meaning so local learning will actually introduce assembly specific infinity connections, and that is good for replay of diverse memory patterns. Yeah. So this is summary. So in the first topics, I introduced self-supervised predictive learning in single neuron, and the network of these single neurons can segment and generate and replay probabilistic assembly structure. Okay. So in the second topics, I explain how infinity plasticity determines the properties of activity replay, and what we found was local EI balance enables diverse memory replay. So we have the global EI balance induces more selective memory replay. And I didn't explain this, but maybe you might feel the learning rule for the first model is a bit too mathematical. It is basically derived from machine learning. But with a student, Gaston Seabury, we constructed more biologically realistic learning rule. What we did is we basically modeled back propagating a potential explicitly. And actually, this rule enables surprisingly fast learning of temporal segmentation. So, so if you are interested, paper is available on bioarchives, please check. So, thank you very much.
Thank you very much, Professor Fukai. Very interesting talk, really great work. It's great to see that there's so much detail now entering theoretical models about inhibitory neurons. So there was a lot of resonance in the chat about this talk. So I would start with the Q&A session, I guess, right? So I would read out the questions. I think that's the easiest. So the top voted question was on the role of dendrites in your model. So the question goes somewhat like this. So STDP can already detect spike patterns without dendrites, so essentially in a point neuron model. So what do you think is the role of complex dendrites that we see in hippocampus and neocortex? Or can you make predictions from your model about this?
Yeah, so spike dependent persistence actually also can detect pattern, but so we asked two responses. One is when pattern is continuing, so neurons should continuously respond. And so depending on model, but the STDP often generate response only at the beginning of the stimulus. Yeah, and that is one difference. And also, yeah, although I didn't show biologically realistic learning rule, and yeah, so this actually, so how do I say? Yeah, by explicitly modeling dendritic spikes, no, sorry, back providing action potential, actually learning becomes very fast. So more quantitatively, though I do not know the data here, so for machine learning model, which I showed, so typically neuron requires about 100 times of presentation of repeated pattern. On the other hand, so more biologically realistic model, about 10 times of presentation is enough. And if you consider network of this biological model, actually just 2 or 3 presentations enough.
Mm-hmm.
Yeah. And I don't know whether STDP can also generate that kind of rapid response, but yeah, I think this is a kind of virtue of our model.
Mm-hmm. Okay, great. Yeah, I think I'm not also sure if I understood the question correctly. So Timothy might want to comment on it, or I think the question was really about the complexity of dendrites that we see in the brain. Is it something that your model could make predictions about why this complexity of dendrites with a lot of arborization, with a lot of branches, could make a difference or is useful on top?
Yeah.
Mm-hmm.
Okay, great question. Yeah. I'm sorry that my answer is based on what I'm not showing. Actually,
Yeah.
I'm sorry. So there is even further modeling study. So this model is still two compartment model, but with the same student. So he is now in Germany.
Mm-hmm. Okay.
And so our next project, we introduced a kind of multi-compartment model.
Mm-hmm.
And we also found a particular ion channel distribution is very important to achieve our mechanism. Yeah. And so what the model suggests is actually there is possibility that each branch selectively responds to a particular pattern. And so in that case, connection between dendritic branch and some soma is established for a particular branch. And then neurons may be able to switch between different selectivity by inhibitive plasticity or something. But this is half of what I said is just speculation. We still have to prove it by explicit modeling. Yeah.
Okay.
Okay, if I can, thanks for your answers, Tomoki. Great talk. Yeah, about the fact that STDP learns with many repeats, I'm not sure I fully agree. In fact, depending on the parameters, you can get it to learn fast. But about the fact that you get, a sustained response during the whole repeating pattern, this is a big difference, I think. With STDP, you would only respond to the first part of the repeating pattern. So, great answer.
Yeah. Thank you for comment. And I forgot to answer. So for the pattern detection, STDP also, of course, works. But in the main topic, so I wanted to learn probability distribution. And that requires at least mathematically predictive learning. It already contains the concept of probability distribution. Therefore, learning of probability of sensory stimuli is no, I don't say it's trivial, but it is expected. But STTP itself doesn't have any. I don't say, but
I agree. It's another difference then. Yes.
Yeah.
Thank you.
So let's maybe move on to the next question because
Sure.
there were many.
Of course.
So the next question was on the role of replay, and if you can comment on if there are other brain structures besides hippocampus that show replay and that can be where your model can make predictions about, essentially.
I see. Yeah. So I think, I think there was evidence from motor cortex, about replay, but I'm not sure. But actually, I'm sorry, my comment is again based on ongoing study. So we actually, I have a collaboration with a human experimentalist. So what I asked was, so I asked patient to remember motor sequence or chunking of motor sequence, and we recorded EEG. And actually, we could get some evidence that during resting state, this pattern, motor pattern, is replayed in the cortex. Around the motor cortex. So. So at least there are, I think, basically learning, all types of learning probably require replay of activity. Even song learning of songbird learning, I think if you deprive sleep, then the performance of learning drops very significantly. So although there is no, I don't know whether there is explicit evidence, but I think learned pattern, motor pattern is replayed during sleep.
So one thing that was mentioned in the chat now is retinal waves during development. So retinal waves, which is probably not, it's far away from motor cortex. So could this also be something that is, I don't understand the question actually, to be honest, what exactly they are pointing at, but I guess if this is also something the model could provide a deeper insight into what the role of retinal waves would be during development. But I think
retinal, so what they mean is retinal, but during development, right? So I don't know whether this retinal wave represents information about sensory stimuli. I'm not very familiar to vision system.
Yeah, I mean, I think we can also go to the next. Yeah. Maybe the formation of the priors in your model could be something that is happening during development, I guess. But I guess the next most voted is also on this Bayesian priors. So how far is our understanding of how spontaneous activity generating neurons approximate the Bayesian prior of stimuli and how they generate the activity?
I think so. There are, I think, evidence that the single neural activity actually or firing rate represent the probability of stimuli. I forgot where it is. Somewhere maybe prefrontal cortex or parietal cortex. I thought, about visual stimuli. So single neuron level evidence actually exists. So if the probability becomes twice large, then firing rate becomes twice large, something like that. But I don't know whether there is evidence from population activity. Yeah. So my model is based on cell assembly. Yeah. So I have to count all spikes from cell assembly responding to the same stimuli. Then probability is accurately represented. But honestly speaking, I do not know whether there is experimental evidence for that.
Yeah, I guess it's also very hard to collect this data,
Yeah.
right, experimentally. Yeah. Okay.